Hi, everyone. Thanks for taking the time to join us this morning. My name is Anne-Marie Garcia, and I'll be your moderator. I'm on the business development team at E4H, and I'm very excited to be hosting this session today. On behalf of E4H Environments for Health Architecture, I'd like to formally welcome you to today's webinar, Modular Facilities for Accelerating Vaccine Development, Planning Strategies for Adapting Multi-Therapeutic cGMP BSL-3 Cleanroom Modules for COVID-19 Response and Cell Therapy Development. In the crucial race to develop and produce a vaccine for COVID-19, one of the longest timelines on the vaccine production cycle is the construction of manufacturing facilities. In today's webinar, Jeff Schantz will address this issue and offer modular solutions to significantly reduce the construction timeline. Before we get started, I'd just like to go over a few logistical items. First, we'll be recording today's live webinar, and it will be available on demand shortly after this session. An email will be sent to you when the recording is available. Second, there'll be a Q&A session after the presentation concludes. If you have a question for our speaker, please feel free to send it through the Q&A tab in the toolbar at the bottom of your screen at any time during the presentation. You can also use the upvote feature if someone else has posted an interesting question that you'd like to see answered today. If we don't get to your question within the time we have together today, we'll be sure to follow up afterward. Lastly, the session is approved for 1 AIA continuing education credit. Should you wish to receive this credit, please send your AIA number to my email address listed on the screen. For those of you who may not be familiar with us, E4H Environments for Health Architecture is a 100% healthcare and health sciences design firm with 8 offices nationwide. E4H is committed exclusively to supporting the discovery of life-changing therapies and improving health outcomes through inspired design. Our team of thought leaders and visionary designers create flexible, state-of-the-art facilities that inspire medical research and enhance the lives of patients and caregivers. I'm very pleased to introduce today's speaker, Jeff Schantz, Director of Health Science and Technology. He's a recognized thought leader with over 30 years of experience designing landmark facilities for college and universities, research institutions, federal government agencies, biotechnology, and pharmaceutical clients all over the world. Jeff has a long list of esteemed clients, including NASA and JPL, Regeneron, Jackson Laboratories, UT Southwestern Medical Center, and the Wadsworth Center. In his downtime, Jeff enjoys being quarantined with his wife, baseball, cooking, and spending time with his golden retriever, Suki. So without further ado, let's go ahead and get started. Jeff, over to you.
Thank you, Anne-Marie. First of all, thank you everyone for joining us today. I hope that you find this informative and interesting. So I got the idea for doing this after reading this article in the New York Times, which was published last Friday and appeared in the Sunday Times. And what the article was asking was, why does it take so long to produce a vaccine? So one of the things it did was it stated the problem. And a traditional typical vaccine cycle could take an entire generation. And one of the things that I noticed in all this was that if you take that typical cycle, the longest timeline on this schedule is the building of factories. So even if we take a best case scenario, which is the idea that we would have a vaccine in 2021, you're essentially taking something that takes a decade to a generation and compressing it into a 2-year schedule. So because of that, how do we go about shortening that particular time frame? The other thing that the article took a look at was a couple of vaccine development scenarios. And what I found interesting about this was it made a couple of assumptions. The first one was that if we assume that we already understand the coronavirus, in other words, enough research has been done to date to understand how the virus works and what its pathology is, if we did that immediately, and we still tried to shorten it, we might have a vaccine on this time frame by 2032, which again is a long time. So if you take a look at what options are there, even if we start the trials early and we don't wait for the academic research, you're still looking at a timeline to build factories of almost 5 years. So what happens if we start preparing those factories now? Even if we start saying that we're going to start building the factories early, in the current way of doing things, we still might not have factories for 5 years. So how can we actually start preparing factories now and build those factories in such a time frame that we can cut the production of the vaccine by years? And so if you look at the way that this schedule looks, what they're really talking about is building the factories now ahead of the finishing of the phase 3 and 2 trials, so that when manufacturing needs to become available after phase 3 trials and there's approval and distribution, we can be able to react to that. So the next question is, how likely is it that we will be able to produce a vaccine in 18 months? This page was part of a study that was done by the Massachusetts High Technology Council, which includes many of the biotechs that are involved in this effort. And as you can see here, what they were looking at in terms of vaccines and in terms of timeframes is the two modalities that they're talking about in terms of the early candidates progress are those vaccines that fall in the mRNA space or the DNL viral vector space. And the vaccine development timelines that they look at, if we do it in the traditional way, you're looking at something that's going to be more than 2 years. So really what we were looking at was what can be accomplished in that 18 months. And it is likely that we could develop a vaccine in that 18-month time frame. But what that really means is it really means that we would understand enough, we would have enough of a proof of concept that a vaccine might actually be available. And many of the companies that you see there on the right side of the page are actively involved in human trials already. So we're starting to move already into human challenge trials. But the real question is which method is going to actually work? In other words, there are a number of different vaccine platforms. The 5 listed here are virus-like particles, synthetic viruses, DNA recombinant platforms, RNA platforms, which is really simplifying some things, or viral vector. And in using any one of these platforms, the thing is what type of vaccine will we be producing? Would we produce it in eggs, which is the traditional method? Would we do something that is cell-based, where you're growing viruses in bioreactors? Or would we be doing something that's more recombinant? And that involves an entirely different type of stream because of the need for the linear DNA plasmids. So since we are at a stage of development now where we're actively seeking how to produce a vaccine and we don't know which method works, how can we be sure that the infrastructure that we're going to put up would actually be able to handle whichever production method comes around? So this prompted the question to me, which is how can we help? How can those of us in the AEC industry help the pharmaceutical companies, the researchers, government agencies, institutions accelerate the design and construction of the vaccine production cycle? Our role in this is we can't do a whole lot to affect what might happen in the research side of things, but we can certainly do a lot to help affect the manufacture, the development, the packaging, the distribution, and what happens to the vaccine once it's actually discovered. And so what this thought experiment really is about is how can we harness the talent, knowledge, and capacity within our industry to help your industry achieve this. What we focused on early on was what types of facilities might be needed to do this. Because we're dealing with a pathogen that really doesn't have a vaccine or a cure yet, we know that we're going to have to work in a biosafety environment. And depending on how you read the guidance from the CDC, it would be at level 2 or 3. But we also need to meet the cGMP requirements for injectables for the FDA. And then because we're not sure of which method is going to be the one that's going to prevail, the only way to protect the investment and make it useful is to make it so that the facility solution that we would offer, the modular solution that we offer, would be able to support any one of these therapeutic work streams. So, as we look down those requirements, you're talking about that biocontainment and that classification. We have to look very quickly at the airside classifications, because if the vaccine is going to be available to everyone in the world, it would have to meet the international standards. The cGMP facilities requirements, we would have to not only meet FDA, but also what would be required by the EU and the EMA, and then beyond that, ISO requirements. Then we would need to be able to understand what those operating protocols and SOPs would be. And then finally, in order to make the facilities available to go active live and start producing vaccine that could be distributed, because we have to make sure that they can be validated and that we have compliance to all of those regulations. This is one of the reasons why the acquisition cycle for the facilities takes so long. So why would we focus then on doing a modular solution, and why multi-therapeutic? There's a couple of reasons why. The first one is that suitable manufacturing sites already exist. And whether that's an existing high bay warehouse or a multimodal building, or even a vaccine production facility that is already in existence, let's say that you have a manufacturing line that can do the high speed bottling and fill finish line, you may not have the actual facility that you need to produce the vaccine given the methodology. So we think that the suitable manufacturing sites exist, and that speeds things up. The second reason why we think that the modular is a good idea is because it's a systems of systems approach. Because we can create the environment in which the instruments would go into for the production of the vaccine, we would be able to front-end load the manufacture of the modulars, but we could also pre-certify, pre-validate, and pre-test those environments before they ever showed up on site. We can deploy the pre-manufactured multi-therapeutic units very quickly, and because of the pre-validated processes and equipment, when we go to bring it all together, we can bring it together at a very fast schedule. The next thing is that we can look at the way that the scale-up process works, and we can do it in an additive way. As everyone knows, when you start doing biologics, whether it's a monoclonal antibody or a vaccine scale-up, the devil in the detail of making it work for creating millions of doses is that when you start to scale up beyond smaller batches and you start to go to larger batches, there are always issues that you run into in the production of things. So one of the things that may help scale up and speed up the process would be to run smaller batches. Instead of trying to go from 1 to 2 liters to 10 liters to 100 liters to 2,500 liters, maybe the thing to do is to make it work at 100 liters or make it work at 20 liters and multiply those things out. So that instead of trying to get to a huge production cycle, we just get to smaller batches and more of them. And this may be important in the way in which we go about this. The next idea that came to us was that following this model, we would be able to do a distributed independent production chain rather than a global supply chain dependent vaccine delivery. And the reason why we think this may be important is because as we witnessed in the ramp up of the pandemic with PPE and ventilators and medical supplies, the global supply chain broke down. And it broke down not because the global supply chain wasn't capable, it broke down because of other reasons, either socioeconomic, political, or technological. And the fact that most airplanes weren't flying, that became a problem. So one of the things that we think we might be able to do is once the vaccine is done and we've created these modulars, that they would be able to be set up anywhere and in multiple locations. The other reason for going to multiple locations is if we have only a single facility worldwide for producing all of the vaccine, if that facility goes down for any reason, including a terrorist event, a bio-terrorist event, a lack of supply, a failure of that facility, a loss of electricity or power, if any of those redundancies happen, we would be unable to produce vaccine. So we believe that this would be a pathway to do something that was more distributed. And then finally, the idea that the modulars could accommodate any process, regardless of what that production cycle is to our advantage. So where do we start? We would start by doing some strategic planning up front. The first thing is to do some risk assessments. The risk assessments need to be done mostly around biosafety and regulatory issues. In other words, how do you protect the people, the product, and the facility? How do you meet all of the regulatory requirements involved in producing vaccines? But if the vaccine includes any potent compounds, for instance, things that might be produced that are not only vaccine related, but prophylactic related, are those things involved? The next thing is to do some due diligence about site assessments, building assessments, and then around commissioning. For instance, if we identify a site that would be suitable, a building that is already in existence, or a production facility, what utilities exist there? What are the characteristics of that building? Do we have the dimensions? Do we have the infrastructure available? Then around biosafety, we need to take a look at those operational issues, and more specifically in the current environment, make sure that this is a safe environment for the personnel who work there. The guidance on workplace issues is going to affect how you might do this. And because of the ability to isolate and distance, the modulars may actually help in isolating teams so that we don't have to worry about those issues. Also, we have to take a look at security. And then finally, we go down the strategic planning around the air classification issues, how we do validation master planning, because that's going to be a key to getting started, how that commissioning is going to have to go, especially if it's an existing facility, and retro commissioning, and then how much the operating protocols and SOPs would affect our planning cycle. So let's take a look at how that might unfold. We put together kind of a generic process flow example about how a production for a vaccine might unfold. And it really depends on which method we use. For instance, if we're doing the traditional seeded method, how we do that seed development, and then pre-process around if we do the mRNA or the DNA method, where are those linear plasmids, are they produced? Are they produced here? Are they produced elsewhere? Are there supply chain issues? Then moving on into manufacturing, what is the nature of it? Are we bioprocessing in incubators and bioreactors, or are we doing it in closed cell instrumentation that's capable of doing the cell manipulations? Then how does the QA/QC fold into this? Then once you get past QA/QC, what happens in terms of formulation? What do we need in terms of aseptic processing? Then how do we package and move that final product out? So one of the things that we took a look at in how modulars might answer this is, what would we do within a basic module? What would we do within a scale up? And what would we do within a production environment? The first thing we took a look at was the air classifications comparisons. And the reason why this is important for us is because we need to satisfy all of these things. Now, I'm not going to go through the particulars of any of this, but one of the things that we did take a look at was what the max particles would be, both in terms of the size and in terms of the saturation. And part of the reason behind this has to do with the size of the virus particle itself. So one of the things that we would want to be able to determine very quickly is what air quality would be required to satisfy those environments. And we think that if we stuck to the EU classifications of A through D, we would be able to satisfy most of this across the standards, and that would make the modulars available for use in multiple locations around the world. So the first idea behind this is that we would have a basic three-wall configuration. The reason for the basic three-wall configuration is because we have a biocontainment issue, because we would need to contain where the virus is worked on, at least when it's in its live state, if not in its killed state. We would have to have a process area that would probably need to be at negative pressure. But in order to meet the cGMP guidelines, we would have to have a positive pressure envelope around that. So we'd have to essentially have containment within a barrier. Now, the containment within a barrier could happen in a couple of different ways. The process area itself may or may not be that containment. That containment may be at the equipment or the isolator level, depending on the air grade required and where we need to set that up. So part of the reason for looking at the three-wall configuration is it gives us some flexibility, A, on air quality, B, on where the containment is, and C, on where the barrier is. But the one characteristic that would be that would be common to this and cGMP requirements is the fact that you have a step-down process as you move from unclassified into more classified air and into cleaner environments for the different parts of the production cycle. So that led us to this idea about a basic module. The basic module, if we looked at it as a standalone, and let's say that you were a small research organization that was ready to go into pre-production, or you just wanted to start producing enough vaccine for clinical trials, that this basic module would be a standalone in a figure-8 type of arrangement. Whereas there's a processing area in the middle of the plan. To the left side of the plan, people would come in and materials and product would flow out. And then on the right side of the plan, you'd have a preparation area. And that would be either for inoculation or it would be for the preparation of plasmids or other materials that would go into the processing of it. So as you can see in this particular plan, we're stepping down into our gradations in terms of air quality, but the processing area would be dependent upon creating that overall environment for the equipment, but moving equipment in there that would be specific to the process depending on whatever it was. And that the process for the material flow would really be from the left-hand side to the right-hand side. So that if raw materials are coming in through that prep room in that airlock area, the processing would happen that would turn it into our material that we would use for the virus creation. And then from here, those materials and waste would flow out to some other process area. And that would just be our basic module. If we're looking to scale that up, and let's say we're looking at our, again, a scaling up of our 3 basic activities, is we have the cell culture and the seed development, we have the manufacturing or the cell processing, we have some formulation or fill finish. So if we had to take our basic module and scale it up to a facility that was, say, capable of producing 1,000 to 10,000 doses, that we would be able to take this and put 3 modulars together. We would be able to link those modules using pass-through positive pressure boxes, where the product would flow from that inoculation and cell prep through the cell culture and seed development into the manufacturing area, into a formulation area that could possibly do some fill finish for a limited amount of production, and then some labeling area before it would move out of there. And again, this would be designed to do a limited run of a particular vaccine. So for instance, if we're looking at operations where we're doing different generations of that vaccine, let's say the first one fails, but we learned enough to do another one, we could either repurpose this or we could build another scale up next to it where we could continue it so that we wouldn't lose any time in that development. And then as we move from a scale up into a production facility, that we might go to scale like this, where the modules themselves, you have a string of modules that would do the cell culture or seed development, a string of modules that would be available to do the bulk manufacturing or cell processing, and then a string of modules that would do the formulation, maybe the filling. And then you surround those with all of the other things that you would need for a factory. So all of the process systems, the QA, QC would move outside of the modules. A major gown in and gown out would be available to get you in, and then you would do another level of PPE when you entered. You could have a separate packaging and labeling, and then as you get to production and ramp up, there would be an external cGMP warehouse so that if your vaccine needs to be stable in a cold storage, we could have cold storage, a shipping and receiving area, and a materials processing area. So as you can see, we can go from that basic module to a ramp-up situation through scale-up and through production. We think that this may be important because, as everybody knows that's trying to produce one of these things, sometimes when you do vaccine production, the first ones fail. So one of the things that we might have to do is we might have to fail faster in order to get a vaccine to market and out to distribution. So let's look at a few examples. And the examples you see on the screen here are courtesy of our friends at GCON. They're a production house for manufacturing the modules. And one of the things that we think we can do is, you can see here on the top picture, These are modules that have been lined up in a warehouse situation where the infrastructure, the utilities, the electricity are all being distributed from the ceiling on down. And then the airside components in this particular one are on the roof of the building and are being ducted down into the modules. We consider the module to be an instrument. So the instrument that is the module can be validated on site. And then once you are done with this particular production cycle, they could also be repurposed. So what this does is it gives you a lot of flexibility in terms of how you acquire the infrastructure, but flexibility in how you redeploy it and how you go through putting in the multiple therapeutics. For instance, if you're done producing vaccine in it, it could be used for other cell processing by being repurposed. Because it's scalable and because these can be cleaned and repurposed, we think that these can go back into a research situation where you can use them for other things. So for instance, in the example you see here up on the top, while there's a lot of airside stuff coming down and piping down, coming down from the ceiling, from the roof, the actual distribution of all of the ductwork is internal to the module. So let's say that you start off with modulars in the beginning that are not specific to a particular process, but enough gets learned in the production of the vaccine that we can make them specific. The modules themselves can be pre-manufactured with all of that infrastructure in place, so that when you move it into the facility, you're literally hooking it up to the airside components and the infrastructure side components. So how do we actually speed this up? You need a procurement strategy. One way we can do this is you can do it in the traditional design, bid, build method, but that tends to take the longest. But that may have to be an option for certain agencies, government agencies who procure these things. But it establishes the cost at the latest date and has the longest acquisition time. Another way to do this would be to do it as design build. And this is available to a lot of government agencies, but it's also available within the industry. And this would have the ability to cut the time frame down significantly by going from preliminary drawings into detailed drawings. What we've been looking at with a number of our clients is this idea that we would do a basis of design that would serve as a bridging document. The basis of design for the bridging document would establish the cost a little bit later, but when it comes to the actual procurement, it would allow you to go to contract with a number of different form of contracts, whether it's a design build, whether it's an EPCM, whether it's a CMAR, or whether it's an integrated project delivery. The bridging documents would allow the design build team to pick them up and turn them into a permit set by taking the BIM models and going directly into fabrication. That would also make the construction of the modules available to go into right away, so that you could do all the enabling work, you could get the field installations underway, and even do some pre-validation for the acceptance. So the bridging document strategy, we think, would be the fastest go-to-market strategy to get you up and running. So let's talk about a few takeaways. So one of the things I want to encourage everyone to think about, and especially those of you who are in the research and the pharma world, understand that in the AEC industry right now, the expertise and the capacity is available. We have people who are available. I know that I've talked to my friends in both the construction industry, on the engineering side, on the validation side, on the commissioning side, that the capacity is available. If you want us, we're there. The second is that we have a method now for the faster procurement and the production and the construction of these using the modulars, and we think that that would be a way of getting us to vaccine distribution much faster. There's a lot of flexibility in this. The flexibility is not only in the way that we would deploy them and construct them, but also in the contracting, the using, and the repurposing of the instruments. And because these are modulars, there is a cost recoverable investment. For instance, there are some suppliers out there who will lease you this equipment. You don't need to have to buy it right away. So if funding is an issue, leasing might be a way to get to it faster. The multi-therapeutic application gives you flexibility in the production cycle. So if it turns out that one vaccine production method doesn't work, we can always pivot and go to another. The scalability is also of great value. because it would work for development, pilot, and production. It would also give you a multi-platform and multi-method way of doing this. So if the vaccine mutates and we need to go to a different production, the interruption of the stream would not affect the facility design and only would affect the production. And then finally, we think that because vaccines often fail, and they fail early and often, this would allow us to fail faster, but also fail forward, in that we wouldn't have to repurpose a facility, even if we had to repurpose the equipment. So that ends our presentation part. We're going to go to Q&A. Now we saw that there's a few questions that have been posted. And if you have some time to stick around, we'll field some questions and answer the questions that we've got.
Thank you so much, Jeff, for sharing your expertise. That was a lot of great information. And if you weren't able to take notes fast enough, no worries. A link to this on-demand session will be emailed to you when it's available. So moving along, as Jeff mentioned, we're going to go ahead and take some time for questions. If you haven't already, please be sure to use the Q&A button in your toolbar at the bottom of your screen to submit questions. So let's just jump right in. Looks like we've got a question here. Jeff, in your opinion, will the emergence of the COVID-19 crisis permanently change how the FDA approves new vaccines? And if it doesn't, can the USA fall behind other global regulatory markets who seem to have a more aggressive process for testing and approval?
Wow. Okay. Yes, it will change a lot of things because I think that the FDA is trying very, very hard to keep up with the companies that are out there that are moving ahead very quickly. I think that everyone around the world recognizes that if we're going to get any semblance of normalcy back, we have to have a vaccine and we have to have testing technologies. And we all know from experience that those things are probably, they might be dependent on science that's done in the government lab, but the application will come from industry. I think what we will find is that around COVID and around the testing technologies around COVID, the FDA is going to move a lot faster than it normally would. You're also seeing the same from the European agencies as well as those in Asia. I think that everybody is moving as fast as humanly possible. However, the one thing I will say is that FDA's role traditionally has been about safety, 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 and the patient safety that goes with this. The one thing that remains to be seen is if there will be any blanket immunities granted to any of the companies that are doing this. The one thing that I think we do see is both us and internationally, there is a focus on the mRNA and the plasmid DNA therapies because those particular methods don't actually use attenuated or live virus. They only use the particles and they only use the parts that would be effective in stimulating the immune response. Because of that, the likelihood of people becoming infected with the virus as a result of the vaccine is very, very low. And that makes the vaccines, by their very nature of it, to become more, it's a much more safe approach than doing live or attenuated vaccine. So the risk of infecting people from the vaccine, I think, is reduced, and that will probably have the FDA approving those methods much quicker. In terms of the global regulatory markets, I think that everyone around the world realizes that we do need to get to this very quickly. I think that if you look at the efforts in Europe, especially the ones in Germany, they are also around the mRNA or the DNA plasmid production side. I know that the one that's being done by Oxford, they were talking about doing immune response, and I'm not sure I understand whether they're doing mRNA or whether they're doing traditional inoculation, but they've also moved into human trials very quickly too. So I think that that will probably have a big effect on how the FDA behaves.
Great, thanks. The next question, let's see. Can you discuss a little bit about the pricing benefits between stick-built versus modular?
Okay, sure. Traditionally, modulars will cost more per square foot than stick-built will. But I think that in any project like this, you know, you have three things that you can work with. You have cost, you have schedule, and you have quality. In a crisis like this, where we're trying to move very quickly, it's probably more important to pay attention to schedule and quality than it is to cost. Now, the initial capital cost of the modulars will probably not be as good as the stick build, but if you keep in mind that the modulars can be repurposed, they are not a sunk cost. So even if you acquire a modular at a higher per square foot cost, it will have the ability to be cost recoverable later down the line. The second thing to realize about this is that the majority of the cost in this cycle is not in the buildings. The majority of the cost is in the instrumentation, the manufacturing equipment, all of the things, the bioreactors, all of the things, the control systems that go into the manufacturing process. If the modular costs $1, the stuff that goes in it costs $10. So I think the thing to do is, in terms of focusing on the cost, we really need to focus on speed to market more than the actual unit cost, because the unit cost will pay off downstream.
Do you anticipate that these clean room pods will be pre-validated in the shop prior to delivery to the site?
That would be the goal, yes. I think the way to do this is that, depending on which method we would settle in on very early on, would be to build a module immediately. Take that module and validate it right away in terms of its control systems, in terms of its air quality, in terms of its ability to operate that environment. And once that, excuse me, once that module is validated, then you go into production to produce as many of them as we would need.
Great, thank you. Looks like we've got one more question here, and it looks like it was an add-on to the discussion of price benefits. What might be an order of magnitude timeline advantage going with modular for 10 to 15,000 square foot cGMP facilities?
Well, assuming that we have an existing facility available, that you would either stick build that or you would modular build that. I think that the modulars would probably get to market 6 to 9 months before you would be able to stick build it. But the real savings in time, I think, is going to be around the validation cycle. In other words, if we can pre-validate the modulars and get them shipped to a site, get them installed, get them hooked up and turned on, you'd be able to put them in, validate them in place in a very short period of time, and then they would be ready to accept instrumentation and equipment. So even though the savings in the construction period might be 6 to 9 months, the savings in the validation might be even more than that. Remember, every month that we gain on the virus, every month that we gain in getting a vaccine in production, is thousands of more lives saved. So I think that when we talk about how we do this and the methodologies that we have to do this, we may have to suspend our natural inclinations to make the most efficient solution and try to get to the fastest solution. It might not be that the most efficient solution is the most cost-effective either, but I believe that this methodology has the best possibility to offer both speed to market and cost.
Thanks, Jeff, for answering those. It looks like we don't have any more questions as of now, but if a question does pop up after the session, please feel free to email us. Jeff's email and my email will be listed. I believe it is listed on the slide. So we're going to go ahead and close the Q&A session. Thanks again to everyone who submitted. We'll follow up after the webinar. Again, if you have any other questions for Jeff.
And you all have my email. Feel free to just email me, and we would be happy to follow up.
Great, thanks. So on behalf of E4H Environments for Health Architecture, I'd like to extend a special thank you to Jeff Schantz and to the audience. We really appreciate you joining us today, and please be on the lookout for information about our next webinar. Thanks again, stay safe and healthy, and have a wonderful weekend.